Hi, everyone. Thanks for being here. My name is Saurabh Rahurkar, and I'm an assistant professor in biomedical informatics at the Ohio State University College of Medicine. Today, I'm going to talk about how we used an informatics-based, data-driven approach to choose a high-impact target uncovering drug safety gaps in pediatric populations. So let's start with the problem you're trying to address. Pediatric populations have historically been left out of clinical research and are basically what we call therapeutic orphans, not because they're rare, but because the evidence base for how we treat them is so limited. Up to 95% of prescriptions in children are off label, and that's not the fringe, that's a routine. And because of that, drug dosing and indications in pediatric patients are often inferred, not based on direct evidence, but on extrapolation from adult data. That's problematic when we know that children, especially the young ones, have very different pharmacokinetics, disease trajectories, and safety profiles. So the challenge here isn't just a gap, it's a structural blind spot in how we generate and apply evidence in this population. I want to briefly revisit the problem with off-label extrapolation, not from a regulatory lens, but from a pharmacologic risk perspective. Pediatric drug metabolism doesn't scale linearly. Age affects absorption, distribution, metabolism, and clearance, so peak assumptions from adults often fall apart in kids. Second, even with weight-based dosing, pharmacodynamic responses are highly variable. You can't assume the same therapeutic index, which can lead to unpredictable drug responses. And third, ADEs in kids tend to be more severe, not just more frequent, but harder to detect and manage due to vague symptoms and narrow dosing windows. And all this makes off-label use risky by default. That's why we need to have an approach to get dosing information and safety information on medication use in these patients, not just extrapolated models. So there have been some efforts to address pediatric drug safety. From the regulatory perspective, we had two key efforts. There's the Pediatric Research Equity Act, PREA, and the Best Pharmaceuticals for Children Act, BPCA, which were designed to close evidence gaps. PREA mandates pediatric studies, but only when the drug's indication has a pediatric analog. It's more of a requirement than an incentive. BPCA, on the other hand, offers incentives like exclusivity extensions for conducting pediatric studies, but it's voluntary and uptake varies. So these two complementary approaches, one coercive and one sort of providing incentive, were meant to address these studies, and together they have increased or they have improved things somewhat, but the gaps still remain. Over half of drugs prescribed in kids today have not been studied. That percentage is even higher for older drugs and CNS agents like benzonatate or some antidepressants or what have you. So despite progress, the real world signal to evidence gap in pediatrics is still wide. So when we talk about informatics-driven drug safety research, we're talking about using data-driven insights to uncover risks, not just relying on physiology or pharmacokinetics or what we think we already know. Sometimes data points us towards patterns we aren't even looking for, and to explain what that looks like in practice, I'm going to take a little detour and take you to the early days of commercial aviation. So in 1952, the de Havilland Comet became the world's first commercial jet airliner. It was cutting edge for its time, jet engines in the wings, pressurized cabin, sleek modern design, what have you. But in the first year of entering service, three of them broke apart midair with no clear cause. Investigations didn't really know what was causing these things. These weren't budget planes. They had been tested out the wazoo and they had passed all of those safety tests with flying colors, pardon the pun. Eventually, the cause was discovered, metal fatigue. They literally had to recreate crashes in pressurized water tanks to figure out. And basically, repeated pressurization cycles were causing tiny cracks around these square windows, something that no one had thought to test for at that time. It wasn't part of anybody's mental model of failure. As a result, the entire fleet was grounded. The aircraft was redesigned with rounded windows, and modern fatigue testing was born, and this kind of testing became the norm. But here's the key point. This wasn't a known risk. The risk was there, but it wasn't visible. It wasn't something they even knew they did not know. This kind of blind spot, a risk we are not even aware of, is what we're trying to look for in this principle of what we call an uncertainty matrix. So This is the framework that really underpins how we are thinking about pediatric drug safety and why informatics is such a powerful tool in this space. It's borrowed from intelligence analysis and it breaks down risk by two axes, whether we are aware of a thing, the understanding, the awareness, and whether we understand it. So we have the known knowns. This is stuff we all agree on. A good example of this is acetaminophen overdose causes liver toxicity. We know that, it's well understood, it's built into clinical decision support systems, and no one's really surprised by it. Then we have the known unknowns. These are things we don't fully understand, like, for example, the long-term effects of mRNA vaccines, specifically in pediatric populations. We recognize this gap, and in many cases, we're trying to actively study it. Then there is the unknown knowns. These are tricky. This is stuff we already have data to understand, but we haven't connected the dots. It's like, for example, when FAERS contains sporadic signals for some rare ADE that nobody has analyzed or flagged yet. And then finally, we have the unknown unknowns. These are the ones that keep you up at night. These are the unexpected ADs that show in real world use but weren't predicted by clinical trials, preclinical data, or even biologic plausibility. And that is exactly the kind of blind spot we're trying to surface with this framework. So the ground work for this is laid by real world data, and this is basically data collected during routine delivery of care, and that's not meant solely for research. So examples of these include data from electronic health records, commercial claims, or post-marketing surveillance data. For the purpose of this particular talk and the rest of the study that I'm going to talk about, we will look at EHR data from Epic Cosmos, which is a de-identified, nationally representative data on patients, on over 290 million patients. There is Medicare Market Scan, which is a commercial claims database, again, over 260 something million patients. And then we have the FDA Adverse Event Reporting System, which is a post-marketing surveillance database where consumers, healthcare providers, manufacturers self-report ADEs for drugs. So we started with market scan to establish a baseline here, essentially to identify which drugs are being most frequently prescribed to pediatric patients. We filtered for all outpatient prescriptions, meaning we wanted to include these in pediatric age bands and retain drugs with at least 1,000 prescriptions. The cutoff was intentional. It gives us a meaningful volume for signal evaluation, but avoids overfitting the tail. At this stage, we are not looking at safety yet, just volume. This gives us the exposure denominator to contextualize everything else that follows. After identifying high volume pediatric drugs from MarketScan, we cross-link that list with MPRINT knowledge base. So for those who are unfamiliar, MPRINT KB is a curated NLP-driven index of pediatric and maternal drug studies, which includes pharmacokinetic, pharmacodynamic, and clinical trials publications, and has over 670,000 publications focused on pediatric and maternal drugs. So we quantified the number of pediatric relevant publications available per drug and use a threshold of 10 or fewer publications as our marker for evidence sparsity. This gives us a quick signal for where clinical use might be outpacing the literature, which is a classic exposure evidence mismatch scenario. This allows us to prioritize drugs that are not just widely used, but also undercharacterized in pediatric populations. FAIRS was our third data source to evaluate whether high-use, low-evidence drugs were also generating serious safety signals. We limited our analysis to serious ADEs, reported specifically by healthcare providers, and only when drugs were listed as primary or secondary suspect. This basically gives us high-fidelity data and also leans on the very conservative side, so we are unlikely to overestimate or have erroneous findings. Now, in exclusions, we excluded congenital anomalies and ADs in children under 3, mostly because in these cases, the exposure likely occurred in utero or through breast milk, and this signal reflects more maternal drug use and not pediatric prescribing per se. We also excluded suicides and intentional overdoses. These aren't pharmacologic adverse events in the traditional sense, though they can be in some cases. But mostly in this context, we treat them as deliberate misuse events, and including them would blur the distinction between toxicity and intent. Cancer drugs were excluded as well, not because they lack ADEs, but because the clinical context is fundamentally different. In most oncology cases, the benefit risk math is already clear and pharmacovigilance is tightly managed. And these patients are also very closely monitored. And just to be thorough, we also excluded supportive agents that are used to manage the side effects of chemotherapy since they kind of operate in the same risk ecosystem. So what we are left here with are unexpected serious ADEs in general pediatric population, the kind that might otherwise get missed in targeted clinical trials or legacy surveillance systems. Again, in both the inclusion and the exclusion criteria, we lean heavily in the conservative side just to not overestimate the effects that we're seeing. So to extract signal strength from pairs, we used disproportionality analysis, specifically proportional reporting ratios or PRRs. Most of you are familiar with these, but just to level set, PRR compare the reporting frequency of a specific drug ADE pair to the background frequency across the entire database. A higher PRR implies a stronger drug ADE association. In other words, what that means is that the drug being tested for has a higher likelihood of having the ADE compared to the same ADE seen in other drugs. We also looked at the litref variable, which has been available in FAIR since 2014. And what this variable does is that it provides links to articles in the literature if one exists for a reported event. And the idea behind this was in case in the slim chance that the MPRINT KB missed a certain article, this is where we could find it. So this is basically what you're looking at. It's a way to differentiate novel, uncharacterized signals and those already described in literature, kind of like built-in context for signal strength that you're seeing. So once we had all these data sets aligned, we had a prioritization framework that we had to work with. So we aren't just looking for random drug ADA associations. We are focused on drugs that meet a couple of criteria, right? They were frequently prescribed, higher real-world exposure increases clinical and public health impact. They were understudied, meaning they had few or no pediatric-focused publications in the MPRINT KB. They showed a strong safety signal based on disproportionality in fares for serious adverse events. And finally, the FDA drug label, we checked whether pediatric safety was explicitly addressed in these labels or not. This gave us a structured way to home in on compounds that were both high risk and high impact, but still flying under the radar from a regulatory or research perspective. So these are the 25 most prescribed drugs in children sorted by total prescription count. No surprises at the top, where you see antibiotics like amoxicillin, azithromycin, followed by some corticosteroids and antipyretics. I do want to bring attention to this particular drug here with almost 250,000 prescriptions, only because this is the one that we'll be following to the next couple of slides. Now we're looking at the top prescribed drugs cross-reference with the MPRINT KB. We are limiting to only those drugs with fewer than 10 publications in pediatric populations. And right at the top is benzonatate, which we saw had 229,550 prescriptions, but only 9 studies in children. That's fewer studies than drugs that are prescribed 10 to 100 times less often, which makes benzonatate kind of a candidate where we can do a deeper dive, or we should do a deeper dive within this framework. So what we're looking at right now is just a distribution of studies by each drug. And I get it. The x-axis is tiny. I don't expect you all to sort of really read the drug names, but that's where benzonatate shows up. That blue line is the number of studies that exist for benzonatate compared to other drugs, which are all orange. And each line here has the number of publications. So one thing I do want to point out is that the y-axis, which is the number of pediatric publications in MPRINT, is on a log scale, which means every marker increases the number of studies by a factor of 10. So on the extreme left, we have drugs that are heavily studied with almost 10,000 studies. And on the extreme right, we have studies which are rarely studied at all. And with benzonatate, we see that mismatch with wide real-world use, but little evidence. And this is exactly the kind of gap our framework is trying to flag. So we identified over 50 unique serious ADEs associated with benzonatate in FAIRS, and these span a broad range, everything from neurological symptoms to some cardiac symptoms, to make the signal more actionable. And to be able to identify them using real world data, we looked for phenotypes in research and PKB. And based on those, we identified arrhythmias, which includes everything from fibrillation to arrhythmia itself to tachycardia, etc., as ADEs under that category. And then we looked at the seizure category, both of which have validated phenotypes in PKB, which makes our analysis more reliable, so to speak. We also wanted to look at other ADs for which phenotypes did not exist. So for these, we looked at EKG changes and respiratory arrest based on how high the PRRs were. So this is what sort of that looks like. And what we're looking at is the relative frequencies, blue for benzonatate and orange for other drugs. And there's quite a few we're looking at here. But what I'm trying to get at here, mostly with this slide, is that this is how we're getting the PRR, which is the blue by orange. So for example, for seizures, we're looking at 4 by 1 with a PRR of 4 for seizures, which tells us that seizures due to benzonatate are 4 times as likely compared to seizures due to other drugs in FAIRS. So bringing back to our prioritization framework. So we talked about looking at the top 25 prescriptions in market scan. Then we're looking at those drugs which have less than 10 publications in MPRINT KB. And then we're looking at the ADs or drugs which have ADs, which have high PRRs in FAIRS. And that brings us to our unknown in case of benzonatate here. So why zero in on benzonatate? Why does it matter in addition to everything else you've talked about? This is an old drug. This was approved in 1958, almost 60 something years ago, but it's seeing a lot of use today, especially as clinicians look for non-opioid alternatives for cough. And it is a cough medicine, but the way it operates is that it's basically an anesthetic, and its mechanism of action is to block the gag receptors in the throat, which is kind of overkill for cough. So here's the catch. It's being prescribed off-label in ways that do not align with current pediatric guidelines. So if you look at the guidelines for managing cough in pediatric patients, the American College of Emergency Physicians says that the gold standard treatment is no treatment, followed by educating the caregivers about how cough progresses and basically, keeping the patient under observation to see nothing goes south. The guidelines also say not to give any medication. And what we're seeing is that basically benzonatate goes strictly against those guidelines. Benzonatate also has a very narrow therapeutic window, meaning the difference between a therapeutic dose and a toxic one is small. That's risky in any population, especially in kids. The FDA drug label data also has very limited information, especially in the pediatric context. So what that means is that as far as contraindications go, the only contraindication the FDA drug label has for benzonatate is hypersensitivity. There are some warnings for pediatric use, but the label does not itself contraindicate use in pediatric populations. And like I said before, the use has increased in the wake of the opioid epidemic, where clinicians have tried to pivot away from narcotics, and then they have zeroed in on benzonatate as the antitussive of choice. The concern that we're seeing isn't even theoretical. Poison control centers, when they publish the data, have routinely shown benzonatate-related overdoses or benzonatate-related ADs in them. And at least one systematic review from 23 uh, found that the efficacy of benzonatate was questionable, and that the approval of benzonatate in 1958 was based on evidence that does not meet the current standards of rigor. So based on all this work, we currently have a proposal under review. It's called the Pediatric Drug Safety Study for Real World Evidence, or PEDS or WE. This is essentially a target trial designed to evaluate safety signals for benzonatate in children using real world data. So we have 3 specific aims. Aim 1 focuses on using validated phenotypes. We compare pediatric patients who received benzonatate with those who did not, and also compared outcomes between pediatric and adult users. So this part uses market scan data, which is a national commercial claims data set database, which does not include, well, mostly does not include CMS patients. Aim 2 is a validation study. Good science needs replications, and that's what we're trying to do in this aim. We replicate aim 1 analyses in EPIC Cosmos, which is a nationally representative de-identified uh, database of patients, of over 290 million patients in the United States. So this gives us a completely different clinical data source, and this helps us validate findings across two different types of real world data. AIM 3 is focused more on discovery and validation to try to identify additional ADs that we haven't even looked for, and also to look for those ADs for which we don't have validated phenotypes. So here we're looking at EKG changes and respiratory disorders, but also trying to identify other ADs that have not been identified yet. So together, these three aims help us cover the full spectrum from looking at validated phenotypes in commercial claims, replicating those findings in EHR data, and then also looking for previously unidentified ADs. So the Benzonatate study is currently under review. We are also expanding this framework in response to national policy priorities. So the Department of Health and Human Services recently launched the Make America Healthy Again Commission with an initial emphasis on childhood chronic diseases. Among the early targets are antidepressants and stimulants, all of which see high real-world use in pediatric populations. Given that mood disorders like depression and ADHD are among the most prevalent mental health conditions in US adolescents, this focus is timely and well justified. There's a growing concern about overprescription in these classes, especially where evidence for long-term safety and efficacy is limited. So we are now applying this same real-world data, MPRINT KB, and FAIR's framework to antidepressants, a class with a particularly high degree of off-label use in children. So this is structured as a target trial design like the previous study, and it's grounded in real-world data. We begin by identifying the most prescribed antidepressants in pediatric populations using EPIC Cosmos this time. Next, we look at how well these drugs are studied in children using MPRINT KB, focusing on those with limited pediatric evidence. And finally, we analyze serious ADs in FAIRS, and in this case, we include events like intentional misuse and suicides, which are highly relevant in the context of antidepressant use. The target trial framework helps us evaluate real world drug safety where clinical trials or randomized control trials would kind of fall short. So these are the 25 most prescribed antidepressants in pediatric populations. And I want to bring your attention to number 5 here, trazodone, which has over 250,000 prescriptions, just a little less than the next most prescribed antidepressant, which is amitriptyline. Since the drugs that are prescribed for chronic conditions are likely to be studied comprehensively, at least in theory, we set a higher threshold for publications here at about 50 publications. So trazodone in that context is the drug with the most prescriptions with under 50 publications in MPRINT KB. Now, to remind you again, trazodone is the fifth most prescribed antidepressant. In comparison, the top four drugs above trazodone have between 229 to almost 500 studies each. So amitriptyline, which I mentioned, which has just a few thousand more prescriptions in Epic Cosmos, has 229 publications, which is 5 times as many as what trazodone has. So basically, that brings us to the gap in ADs. So we identified over 110 unique serious ADs in FAIRS, and this include everything from cardiac symptoms or cardiac-related ADs to respiratory acidosis and so on. And we are going again with the validated phenotypes for arrhythmia and seizures, also because these are definitely those with high PRRs in the FAIRS dataset. And we are currently in the process of identifying phenotypes for additional ADs, which are highly relevant. So suicides are among those simply because they do show up in the FAIRS with fairly high PRRs. And again, these are a known sort of AD when it comes to antidepressant usage. We also have respiratory disorders, and we also have priapism, some of which are derived from the few studies that we have looked at that have looked at anything to do with safety in trazodone. So again, the prioritization framework, right? So we have top 25 prescriptions in Epic Cosmos. We're comparing those with drugs with less than 50 publications in the MPRINT KB, and then looking at PRRs, which are high PRRs in FAIRS, and that brings us again to the unknown, which is trazodone. Now, why does trazodone matter? Trazodone is a serotonin receptor antagonist and receptor and reuptake inhibitor. It was approved in 1981, so it's not a new drug by any means. It shares several pharmacological features with SSRIs and is frequently used to manage depression in children. It's also used frequently off-label in insomnia, encephalopathy, headache, and also for aggression. And there are 47 studies in the MPRINT KB, but pretty much all of these studies focused on off-label use, be it efficacy or safety. Most of them focus on either using it for headaches or for insomnia, for leg movements during sleep, those sorts of things. Uh, Studies that do exist are very, very small samples. And in general, most of these studies do not focus on trazodone as the primary drug. So oftentimes, these are studies that are focused on other drugs which use trazodone as a comparison. And ultimately, we found three studies which would be considered safety studies, but these were not studies so much as case reports. So again, we have the high use, high PRR ADAs, and low evidence, the trifecta coming into place here. So to close here are the takeaways from our work. We propose an evidence generation framework that helps systematically surface safety concerns in pediatric populations using real-world data. This is made possible by the availability of massive healthcare data like MarketScan and Epic Cosmos. These resources are especially valuable for high-risk and understudied populations like children and maternal populations, where well, clinical trials can be challenging for several reasons. By combining data from multiple sources, we are able to explore the unknown unknowns, surfacing unexpected or understudied drug ADA associations. And here, the data itself helps drive the identification of potential drug safety targets. Ultimately, we hope that these insights will inform future clinical studies, labeling updates, regulatory or peer decisions as well. And I want to take a moment to acknowledge this incredible team behind this work, Dr. Langley, our PI, for his ongoing mentorship and support throughout this project. Dr. Pallavi John Lagadda, she's my postdoc and has been instrumental in every phase of the proposal and framework development. Ada Ouyang is a doctoral student, and she has been doing some great work with the FAIRS, especially in the disproportionality analysis space. Dr. Lei Wang for his contributions in cross-linking the MPRINT KB with the other data sets that we have used in this framework building. Xiaofu Lu, she's another doctoral student who was instrumental in developing the NLP algorithm that powers MPRINT KB. Dr. Jack Chang for his work with the market scan, and finally Dr. Shenrei, who has been doing this work in the opioid space, which served as some of the starting work for building this particular framework. And if you're interested in real world data, digital health, predictive modeling, I'd love to connect. I'm ready for questions. And again, I want to thank the incredible imprint team spanning data science, informatics, pharmacology, and regulatory research. You can connect with me via email, Blue Sky, or LinkedIn. Thank you.